every day, ships cross the Atlantic as if it were fully known. Routes are charted, depths are printed on maps, satellites track the surface within centimeters. It feels measured. But north of Puerto Rico, the seafloor collapses. The Atlantic does not simply deepen, it drops into the Puerto Rico Trench. At its lowest point, Milwaukee Deep lies more than 8,300 meters below the surface, nearly 5 miles down. At that depth, pressure exceeds 16,000 pounds per square inch, enough to crush unprotected metal, enough to collapse anything built for the surface world. Sunlight disappears within the first 1,000 meters. Below that, darkness is permanent. The water hovers just above freezing. Tectonic plates grind slowly along the boundary. For centuries, no human even knew this place existed. Even today, reaching it requires rare vehicles built for a single purpose: to survive descent into conditions that push material to its limit. Scientists once assumed that life, if present at all, would thin into near emptiness. Energy is scarce, food drifts down unpredictably. Pressure alters chemistry itself, and yet, when instruments finally touched the trench floor, they did not report silence. They detected motion. Samples brought back were not sterile. Something lives there. But what survives nearly 5 miles beneath the Atlantic? How does biology endure, where light has never existed? What changes when evolution operates under crushing pressure and cold that never lifts? And how deep do those limits truly go? Tonight, we descend into the deepest Atlantic, not to chase myth, but to examine what was actually found there. But before we begin, if you enjoy calm, late-night journeys into the deep sea, I'd appreciate it if you gave this video a like and perhaps tap subscribe. It helps other curious dreamers find their way to this peaceful little corner of the internet. And if you'd be so kind, let me know in the comments, where are you listening from tonight, and what time is it where you are? It's always a joy to see how far across the world this little sleepy corner of science travels. Now take a slow, deep breath, let your shoulders soften, and together, let us descend toward the hidden abyss of the Atlantic. Imagine the map folding inward until every modern shoreline presses against another, continents interlocked like pieces of a vast geological mosaic. In that distant configuration, the lands we now separate by oceans were united within a single supercontinent known as Pangaea. What would one day become the Atlantic Basin lay hidden beneath forests, river systems, and mountain chains that stretched uninterrupted across hemispheres. Beneath that immense landmass, the mantle was never still. Heat rose slowly from deeper layers of earth, spread laterally, then cooled and sank again in broad convective loops. Over tens of millions of years, those currents thinned the crust from below. The surface responded gradually. Long fractures opened across Pangaea, forming rift valleys that widened as magma welled upward to occupy the space created by separation. Volcanic activity stitched new material into the gaps, dark basalt cooling into fresh oceanic crust. As the rifts expanded, seawater advanced into the low corridors between dividing plates. A narrow sea emerged, lengthening and deepening with time. Along its center, a continuous volcanic spine took shape, the structure now known as the Mid-Atlantic Ridge. This ridge remains one of the most extensive mountain systems on Earth, though it lies submerged beneath kilometers of water. Here, plates diverge at a measured pace, often only a few centimeters each year, yet over geological ages, that motion is enough to create an ocean. The Atlantic Basin is therefore a record of steady expansion. New crust forms at the ridge axis and then migrates outward on either side, cooling and thickening as it travels. Because this spreading has been active for a shorter span of Earth history, than similar processes in older basins, the Atlantic seafloor is comparatively young. In contrast, large areas of the Pacific Ocean preserve crust that has endured far longer before descending back into the mantle at subduction zones. The Pacific has had more time to generate, recycle, and reshape its floor, while the Atlantic is still in an earlier chapter of that cycle. This difference in age and tectonic setting influences the ocean's structure. The Pacific is encircled by active margins where plates converge, producing deep trenches and frequent volcanic arcs. Around much of the Atlantic, however, the continents rest on passive margins. Instead of colliding, they drift apart. Without widespread subduction, trenches are uncommon. The seafloor slopes gradually from continental shelves into broad abyssal plains, forming a basin defined more by spreading than by descent. Yet the Atlantic is not entirely uniform. In the northeastern Caribbean region, motion becomes more complex. The North American plate moves relative to the Caribbean plate in a pattern that combines sideways sliding with compressive force. This interaction does not mirror the steep symmetrical subduction typical of the Pacific margins. Instead, it creates localized bending and deformation within an otherwise spreading ocean. Through this oblique convergence, sections of the North American plate are flexed downward beneath the Caribbean plate, carving a deep trough in the seafloor. The surrounding Atlantic continues to widen along the mid-Atlantic ridge. Yet here, the crust experiences a different stress regime. Growth and compression coexist within the same basin, guided by mantle circulation and plate motion that began when Pangaea first fractured. The Atlantic today carries the memory of its origin in every kilometer of seafloor. Its central ridge continues to generate new crust, while its margins largely remain quiet and stable compared to the fiery perimeter of the Pacific. Only in select regions do plates press together strongly enough to produce pronounced depth. The ocean that now separates continents is still widening, its boundaries shaped by the same gradual forces that once pulled a single landmass apart and allowed water to fill the space between drifting worlds. North of the island arc of Puerto Rico, the seafloor lowers almost without warning. The broad Atlantic plain gives way to a narrow depression that stretches in a long arc beneath the open ocean. From above, nothing on the surface reveals its presence. Waves pass freely overhead, unaware that below them, the crust folds into one of the deepest places in the Atlantic basin. The Puerto Rico Trench extends for roughly 800 kilometers, curving east to west along the boundary where tectonic plates meet. Its width varies, narrowing in some sections to less than 100 kilometers before widening again. The trench does not form a perfect line, but a complex series of troughs and ridges, shaped by uneven forces within the crust. Sediment gathers along its slopes, softening its contours, while deeper sections remain sharply defined. At its lowest measured point lies Milwaukee Deep, descending more than 8,300 meters below sea level. This basin rests within the trench's western sector, where the downward bend of the North American plate reaches its greatest depth. The surrounding walls rise steeply, enclosing a narrow floor that holds the coldest and densest Atlantic waters. The pressure there exceeds that of most of the ocean, rivaled only by the Hadal zones of the Pacific. For much of human history, the trench existed only as an absence on maps. Early depth soundings taken by weighted lines hinted at unusual depth north of Puerto Rico, yet the measurements were sparse and uncertain. In the 20th century, echo sounding techniques began to trace a clearer outline. Pulses of sound traveled downward, struck the sea floor, and returned with subtle delays that revealed contours hidden beneath kilometers of water. As technology advanced, multi-beam sonar systems produced more detailed bathymetric images, transforming the trench from a shadow into a mapped structure with measurable form. Cold War naval surveys contributed further precision. Submarines navigating deep Atlantic routes required accurate charts of underwater terrain. Data gathered for strategic purposes quietly enriched scientific understanding. Over time, repeated surveys refined the estimated depth of Milwaukee Deep, narrowing uncertainty until consensus settled around its present measurement. The trench became not just a theoretical depression, but a charted landscape with coordinates and relief. Its origin differs from the sweeping subduction systems that dominate the Pacific margin. In those regions, oceanic plates plunge steeply beneath continental or other oceanic plates, forming long chains of trenches aligned with volcanic arcs. The Puerto Rico Trench developed under more oblique conditions. Here, the North American plate moves westward relative to the Caribbean plate, with motion that combines sideways sliding and downward bending. Instead of a simple head-on descent, the interaction creates a complex deformation zone. This oblique convergence produces uneven compression. Portions of the North American plate flex downward, while adjacent areas experience lateral shearing. The result is a trench that is deep yet geologically intricate, lacking the continuous volcanic arc typical of many Pacific examples. Earthquakes occur along this boundary, marking the slow transfer of strain through the crust. The trench's form reflects this interplay, shaped by forces that are neither purely divergent nor fully convergent. Seen within the wider Atlantic, the Puerto Rico Trench stands as an exception to the ocean's general pattern of spreading margins. It occupies a place where motion shifts direction and the seafloor yields to pressure. Its walls descend into stillness, holding waters that have traveled far from the central ridge. Though hidden beneath the surface, the trench remains an active feature of a living planet, carved by tectonic movement that continues in quiet increments beneath the passing sea. Long before digital charts and glowing screens, depth was measured by hand, one length of rope at a time. Sailors stood at the rail and lowered weighted lines into the open Atlantic, waiting for the moment the lead touched bottom. Knots tied at intervals along the rope-marked distance. The line would descend through cold layers of water, swaying with currents, until slack signaled contact with the seafloor far below. These early soundings were patient and imprecise. A single measurement could take hours. Storms interfered. Currents dragged the line at angles, distorting results. In deep water, the rope itself grew heavy with its own weight, stretching under strain. Yet even with such limitations, scattered readings hinted at dramatic changes in depth north of the Caribbean. Where charts once suggested uniform seafloor, unexpected numbers began to appear, larger than anticipated. By the early 20th century, a quieter and more reliable method emerged. Echo sounding replaced rope with sound. A pulse was sent downward through the water column, striking the seabed and reflecting back to a receiver. The time required for the signal to return could be translated into distance based on the known speed of sound in seawater. What once demanded hours could now be measured in seconds. Continuous profiles became possible as ships moved steadily forward. As technology advanced, sonar systems improved in sensitivity and precision. Single-beam echo sounders traced narrow lines across the ocean floor, revealing contours previously hidden in darkness. Overlapping transects slowly assembled a broader image. Patterns of slopes and basins began to take shape on bathymetric charts, though large gaps remained between survey tracks. During the mid-20th century, geopolitical tension quietly accelerated ocean mapping. Submarine navigation required accurate knowledge of underwater terrain. Bathymetric surveys conducted during the Cold War gathered detailed depth data across vast portions of the Atlantic. Though many missions were classified at the time, their measurements contributed to a more refined understanding of deep ocean structures. The trench north of Puerto Rico became clearer in outline as data accumulated from multiple passes. A further transformation came with multi-beam sonar. Instead of sending a single, narrow pulse downward, these systems emitted a fan of sound waves that covered a wide swath of seafloor. Each return signal was processed to create a high-resolution map of depth and slope. The ocean floor, once represented by sparse lines, emerged as a textured landscape with ridges, escarpments, and basins rendered in detail. Subtle variations in topography could be detected, allowing scientists to identify the deepest depressions with increasing confidence. Accurate measurement required careful calibration. The speed of sound in seawater varies with temperature, salinity, and pressure, so instruments had to account for changing conditions through the water column. Sensors lowered from research vessels recorded these properties, ensuring that sonar calculations reflected the true distance to the seabed. Repeated surveys cross-checked earlier results, narrowing uncertainty. To confirm the depth of the deepest basin, direct observations were eventually paired with sonar data. Specialized submersibles and remotely operated vehicles descended to the trench floor, carrying pressure sensors and altimeters that verified readings taken from above. The convergence of independent methods strengthened the measurement. The value assigned to the deepest point was not the result of a single sounding, but of accumulated evidence gathered over decades. Through rope, sound, and advanced instrumentation, the abyss was gradually translated into numbers. What began as isolated hints became a coherent map. The seafloor that once resisted measurement now lies charted with remarkable precision. Yet, even with modern technology, each survey carries the quiet acknowledgement that the ocean remains vast, and that every depth recorded is part of an ongoing effort to understand a landscape concealed beneath kilometers of water. Pressure begins quietly at the surface, almost unnoticed, a gentle weight of air resting against skin. Step into the sea, and that weight changes character. Water, far denser than air, presses inward from every direction. With each descent of roughly 10 meters, the force increases by about 1 atmosphere, adding another layer of compression around anything that sinks below. At modest depths, the change is manageable. Divers equalize the pressure in their ears, and specialized equipment supports breathing, yet the increase continues without pause. At 100 meters, the surrounding force reaches about 11 atmospheres. At 1,000 meters, it approaches 100 atmospheres, a load equivalent to several tons pressing against every square meter of surface. Now consider a descent beyond 8,000 meters. At such depth, pressure exceeds 800 atmospheres, translating to more than 16,000 pounds per square inch. This force does not push from one side, as wind does against a wall. It surrounds completely, seeking any weakness, any microscopic flaw. Steel, aluminum, glass, all materials behave differently under such uniform compression. Some deform, others fracture suddenly when their structural limits are reached. The human body, adapted to life near the surface, cannot endure these conditions unprotected. Our tissues contain air spaces within lungs and sinuses. As pressure increases, those spaces compress. Without a rigid barrier to maintain internal equilibrium, organs would be crushed and gas dissolved into blood at dangerous concentrations. Even before reaching extreme depth, high pressure alters the chemistry of breathing gases, leading to nitrogen narcosis or oxygen toxicity. Beyond certain limits, survival is not a matter of endurance but of physics. To explore such regions, engineers design vehicles that resist collapse by distributing stress evenly. Deep submersibles often employ spherical pressure hulls because a sphere minimizes stress concentrations. Thick walls of steel or titanium contain a small interior cabin where pressure remains close to 1 atmosphere, allowing occupants to breathe normally. The trade-off is scale. As depth increases, hull thickness must increase as well, reducing interior space and increasing weight. Materials are chosen not only for strength, but for predictability. Titanium alloys provide high strength to weight ratios and resist corrosion in seawater. Acrylic viewports, used in shallower submersibles, become impractical at extreme depths, replaced by smaller quartz or specialized glass windows designed to withstand compression without shattering. Each component, from bolts to seals, must be engineered with precise tolerances, because even a minor defect can propagate rapidly under hundreds of atmospheres. Uncrewed vehicles face similar constraints. Remotely operated vehicles rely on pressure-compensated housings filled with oil, allowing external pressure to equalize across components. Electronics are sealed within rigid chambers. Syntactic foam, composed of hollow glass microspheres embedded in resin, provides buoyancy while resisting collapse. Its structure allows it to maintain integrity where ordinary foam would implode. Despite these innovations, structural limits remain absolute. Every material has a compressive strength beyond which it fails. Engineers calculate safety margins, testing hulls in hyperbaric chambers that simulate deep ocean conditions. Even so, descent to hadal depths carries inherent risk. The deeper a vehicle travels, the smaller the margin between operational pressure and material failure. Pressure at 8,000 meters is not an abstract number, but a continuous physical reality, acting uniformly and relentlessly. It shapes the design of every bolt and beam intended for descent. It defines the boundary between surface life and the abyss, between environments where biology evolved freely and regions accessible only through careful engineering. In that immense compression, the ocean reveals a simple truth of physics, that force increases with depth, and survival depends on understanding how to balance that force with structure strong enough to endure it. Light begins its journey at the surface in rippling bands, bending and scattering as it passes from air into water. For a brief distance below, the sea remains luminous. This upper layer, often called the sunlight zone, extends to roughly 200 meters. Here, photons are still abundant enough to support photosynthesis. Microscopic phytoplankton drift within reach of the sun's energy, forming the base of vast marine food webs. Colors appear familiar in this realm, blues and greens softened by motion, shadows still defined. As depth increases, brightness fades gradually rather than suddenly. By the time the descent reaches about 200 meters, the sea enters what is known as the twilight zone. Between 200 and 1,000 meters, sunlight weakens into a dim cobalt haze. Red wavelengths vanish first, absorbed within the upper layers, leaving objects tinted in shades of blue and gray. Vision becomes less reliable. Shapes blur into silhouettes. Many organisms here develop large, sensitive eyes to capture the remaining traces of illumination. Below 1,000 meters, the ocean passes into the midnight zone. Sunlight no longer penetrates. The water absorbs and scatters the last residual photons long before they can reach such depths. Without an external source of illumination, the environment becomes optically black. This darkness is not the shifting darkness of night on land, but a constant absence, unchanged by time of day or season. It persists without variation. The reason light cannot reach the trench floor lies in the physics of absorption. Seawater contains dissolved salts, suspended particles, and microscopic organisms that interact with incoming radiation. As light travels downward, its energy is progressively absorbed and scattered. Each meter reduces intensity. By several hundred meters, most wavelengths are diminished to near zero. By 1,000 meters, natural sunlight has effectively disappeared. At 8,000 meters, no direct solar energy remains. In this absence, vision ceases to function as it does near the surface. Many deep-dwelling organisms adapt accordingly. Some species reduce or lose their eyes entirely, conserving energy in a habitat where sight offers limited advantage. Others evolve highly specialized retinas, capable of detecting faint flashes produced by living creatures rather than by the sun. Bioluminescence becomes the dominant visual signal in the mid-water above the trench, brief pulses of chemically generated light used for communication, camouflage, or predation. At the trench floor itself, even bioluminescent signals are rare compared to the layers above. The density of organisms decreases, and encounters are less frequent. In this environment, sensory systems shift emphasis. Chemical gradients, subtle vibrations, and electrical cues replace vision as primary channels of perception. Antennae extend outward to sample dissolved compounds. Lateral line systems detect minute disturbances in surrounding water. Life persists without reliance on daylight. The descent through light, therefore, traces not only a gradient of brightness, but a transformation of ecological strategy. In the upper layers, energy flows directly from the sun into living tissue. In the deeper zones, organisms depend on organic material sinking from above or on the faint glow of neighbors. At the trench floor, the sun is an indirect memory, its energy delivered only through detritus that drifts downward over long distances. Above, the surface continues to shimmer beneath open sky. Below, the midnight zone remains unchanged, insulated from cycles of dawn and dusk. The trench lies within this perpetual darkness, shaped by geological forces and sustained by processes that operate without sunlight. In the absence of light, evolution has explored different solutions, crafting forms attuned to pressure, cold, and chemical signals rather than to color and brightness. The ocean's vertical journey from illumination to blackness reveals a gradual withdrawal of solar influence, ending in a realm defined by darkness that never lifts. Far below the restless surface, temperature settles into a quiet constancy, while winds stir the upper layers and seasons alter the warmth of coastal shallows. The deep Atlantic remains almost unchanged. In the trench and across the abyssal plains, water hovers just above freezing, typically between 1 and 4 degrees Celsius. It is not ice, for the immense pressure lowers the freezing point slightly, but it is close enough that movement feels heavy and deliberate. This cold is not a sudden feature that appears only at extreme depth. As one descends from the surface, temperature decreases through a transition known as the thermocline. In the upper few hundred meters, sunlight warms the sea unevenly, creating a mixed layer influenced by weather and currents. Beneath that lies the thermocline itself, a region where temperature drops rapidly with depth. The gradient can be steep, several degrees lost over a short vertical distance, marking the boundary between surface variability and deep stability. Below the thermocline, the change slows. The water becomes part of a vast reservoir that circulates slowly across ocean basins. Much of the deep Atlantic's cold originates in high-latitude regions, where surface water cools, increases in density, and sinks. In the North Atlantic, near Greenland and the Labrador Sea, winter air chills the ocean's surface until it becomes heavy enough to descend. This process forms what is known as North Atlantic deep water, a mass of cold, oxygen-rich water that spreads southward along the ocean floor. Once formed, this deep water moves gradually, driven by density differences rather than wind. It may take centuries to complete its journey through the Atlantic basin. During that time, it remains largely insulated from atmospheric influence. The immense volume of surrounding water buffers against rapid change. Heat exchange with the surface is limited by distance and stratification, allowing the deep ocean to maintain a narrow temperature range over long periods. Pressure plays a subtle role as well. At great depth, compression slightly raises the temperature of seawater, yet this effect is small compared to the overall cold established at formation. The balance between sinking polar water and the slow mixing with surrounding layers keeps the deep Atlantic remarkably uniform. Seasonal cycles that affect coastal waters barely register thousands of meters below. This stability has ecological consequences. Organisms living in the trench and nearby abyss have evolved within an environment where temperature varies by fractions of a degree rather than by tens of degrees. Metabolic rates reflect this constancy. Chemical reactions proceed at predictable speeds. Enzymes function optimally within narrow thermal limits, shaped by millions of years of steady conditions. The thermocline above acts as a boundary, separating two thermal worlds. In the sunlit zone, warmth fluctuates with latitude and weather. Storms can mix the upper layers, redistributing heat. In contrast, the deep ocean remains largely undisturbed by such events. Only major changes in global circulation patterns alter its temperature over geological timescales. Within the trench itself, the water mass is part of this broader deep circulation. Despite the extreme depth and pressure, its temperature differs little from surrounding abyssal waters. The cold is pervasive, but not harsh in the sense of fluctuation. It is a steady presence, a defining characteristic of the deep Atlantic. Above, sunlight and wind continue their shifting patterns. Below, the temperature rests near freezing, sustained by distant polar winters and slow planetary circulation. In that constancy, the deep ocean preserves a quiet thermal equilibrium, one that has endured through centuries of surface change and that continues to shape life in the hidden reaches of the Atlantic. Long before sleek, remotely operated vehicles transmitted high-definition images from the abyss, descent depended on heavy steel, thick cables, and a willingness to enter the unknown with limited certainty. The earliest deep-sea explorers did not begin with the trench itself, but with the broader challenge of reaching depths no human had visited. Their craft were experimental, improvised at times, shaped by trial and error as much as by calculation. Among the most influential designs were the bathyscaphs, free-diving submersibles developed in the mid-20th century. Unlike tethered diving bells, bathyscaphs carried their own buoyancy system, a large float filled with gasoline, chosen because it is lighter than water and incompressible under pressure, provided lift. Suspended beneath this float hung a small spherical pressure cabin built of thick steel, just large enough for a crew of 2 or 3. Inside that sphere, atmospheric pressure could be maintained while the ocean pressed inward with increasing force. Early models faced significant constraints. Visibility was limited to small viewports, often only a few centimeters thick, and carefully engineered to resist cracking. Communication with the surface was unreliable. Navigation depended on rudimentary instruments, with little capacity for precise maneuvering near the seafloor. Ballast systems, designed to release iron shot in order to ascend, required careful control. If a mechanism jammed or released too quickly, the consequences could be severe. Despite these limitations, bathyscaphs ventured progressively deeper into the Atlantic. Test dives evaluated hull integrity under rising pressure. Each successful descent validated engineering calculations and material choices. Failures also occurred. Equipment malfunctioned. Instruments lost calibration under compression. In some cases, external components were crushed, though the pressure sphere often remained intact. These setbacks revealed weaknesses that informed subsequent refinements. As interest in deep ocean science expanded, specialized missions targeted specific regions of the Atlantic basin. Crews descended to abyssal plains, observing sediment fields and benthic organisms directly for the first time. The experience of sitting within a steel sphere while the ocean closed around it reshaped scientific imagination. Depth became not just a number on a chart, but a tangible environment, one that could be entered and witnessed. Improvements followed with each generation. Stronger alloys reduced hull thickness while preserving safety margins. Electrical systems were better insulated against pressure-induced failure. External lights illuminated short stretches of seafloor, revealing textures and movements otherwise hidden in darkness. Cameras were mounted outside the pressure sphere, capturing still images that supplemented human observation. Yet even with progress, early submersibles operated at the edge of technological possibility. Battery life restricted dive duration. Descent and ascent were slow, governed by buoyancy adjustments rather than propulsion. The margin between operational depth and structural limit remained narrow. Engineers calculated compressive stresses carefully, but real-world conditions always introduced uncertainty. Some missions achieved remarkable milestones, demonstrating that human-occupied vehicles could survive depths approaching those of the deepest Atlantic trench. Others were curtailed by mechanical issues that forced early ascent. Each attempt contributed data, not only about the ocean floor, but about the behavior of materials under extreme pressure. Lessons learned from minor hull deformations or valve failures informed safer designs in later years. Through persistence and measured risk, the first submersible attempts established a foundation for modern exploration. They proved that descent into the deepest Atlantic waters was physically possible with careful engineering. Though limited in mobility and endurance, these early craft carried observers into realms previously inferred only from sonar traces. Within their confined steel spheres, surrounded by immense pressure, explorers opened a path toward more advanced vehicles that would follow, building upon both the successes and the quiet corrections of those initial dives. Where early explorers relied on thick steel spheres and narrow windows, modern descent often begins with a quiet launch over the side of a research vessel. A crane lowers a framework of metal and syntactic foam into the sea, and as it slips beneath the surface, control passes to operators seated before screens rather than within pressure hulls. These are remotely operated vehicles, or ROVs, engineered to travel where human presence is no longer required. An ROV carries no crew. Instead, it remains tethered to the ship above by a reinforced fiber optic cable that transmits power and data across kilometers of water. Through this connection, pilots guide thrusters, adjust cameras, and operate mechanical arms with precise movements. The vehicle responds in real time, its actions mirrored on monitors illuminated in a control room far from the crushing depth below. To survive descent into the trench, modern systems rely on pressure-resistant materials refined through decades of research. Titanium alloys are common, chosen for their strength and resistance to corrosion. Electronics are enclosed within sealed housings, designed to withstand hundreds of atmospheres. In some cases, Components are immersed in oil-filled compartments that equalize external pressure, preventing structural collapse. Syntactic foam, composed of microscopic hollow spheres embedded in resin, provides buoyancy without compressing significantly under extreme force. Cameras are central to modern exploration. In total darkness, vision depends entirely on artificial illumination. High-sensitivity sensors capture faint reflections, while digital processing enhances contrast without distorting detail. Housings protect lenses from pressure, and carefully engineered seals prevent water intrusion. These systems must remain stable, despite temperature near freezing and pressure that would crush unprotected glass instantly. Lighting systems transform the immediate surroundings into a visible landscape. Arrays of LED lamps project beams that cut through suspended particles. The light reveals sediment textures, rock faces, and the slow movement of organisms adapted to the abyss. Engineers balance brightness with efficiency, since power transmitted through the tether must support thrusters, instruments, and illumination simultaneously. Excessive heat from lamps is dissipated into the surrounding water, where it vanishes without altering the broader environment. Navigation in the trench requires more than visual cues. Satellite signals cannot penetrate to such depth, so ROVs depend on acoustic positioning systems. Transponders placed on the seafloor emit sound pulses detected by receivers on the vehicle and ship. By calculating travel time, computers determine location with surprising accuracy. Inertial navigation units track motion between acoustic updates, maintaining orientation even when currents shift unexpectedly. Manipulators extend from the vehicle's frame, jointed arms capable of delicate sampling. Sediment cores, biological specimens, and rock fragments can be collected without exposing human occupants to pressure. Instruments mounted on the chassis measure temperature, salinity, and chemical composition, transmitting data continuously to the surface. Each descent becomes both an observation and a recording, preserving information in digital archives. Compared to the first submersible attempts, modern technology allows longer missions and greater precision. Vehicles can hover just above the trench floor, adjusting buoyancy and thrust to maintain position. If mechanical failure occurs, the absence of crew reduces immediate risk. Recovery procedures are planned carefully, yet the stakes differ from those of earlier eras. Through cables, sensors, and resilient materials, the deep Atlantic has become accessible in a new way. The trench remains distant in physical space, yet its contours and inhabitants appear clearly on screens aboard ships that ride far above. Modern deep-sea technology does not diminish the weight of pressure or the permanence of darkness. It works within those realities, translating them into data and images, extending human reach while keeping fragile bodies safely at the surface. Before instruments ever touched the trench floor, expectations formed quietly in laboratories and lecture halls. Depth suggested scarcity. Distance from sunlight implied limitation. For decades, many researchers viewed the deepest Atlantic waters through the lens of constraint, assuming that life, if present at all, would be sparse and slow. One prevailing framework was the energy limitation theory. In surface waters, sunlight drives photosynthesis, generating organic matter that fuels complex ecosystems. At extreme depth, no such production occurs. The trench lies far below the reach of solar radiation, dependent on material sinking from above. This downward drift, often described as marine snow, consists of microscopic particles, fragments of plankton, fecal pellets, and the occasional larger carcass. As these particles descend, much is consumed before reaching the abyss. By the time organic matter arrives at Hadal depths, it has been reduced to a fraction of its original abundance. From this perspective, the trench appeared to be an energetic desert. Limited input implied limited output. Fewer calories available meant fewer organisms could be supported. Models predicted low biomass and minimal diversity, with only highly specialized scavengers surviving on intermittent resources. Some early hypotheses suggested that population density would decline steadily with depth, approaching near sterility at the ocean's deepest points. Food scarcity reinforced these assumptions. Large events such as whale falls, which can deliver concentrated nutrients to the seafloor, are rare and unpredictable. Between such occurrences, organisms must rely on the steady but sparse rain of detritus. In cold, high-pressure environments, metabolic rates are reduced, yet they cannot fall to zero. Life requires a continuous, even if minimal, supply of energy. The trench was therefore imagined as a place where survival hinged on extreme efficiency and patience. Another factor shaping expectations was isolation. The trench forms a narrow depression separated by steep slopes from surrounding abyssal plains. Its depth and topography suggested limited exchange with adjacent habitats. Some scientists proposed that such isolation might restrict colonization, leading either to very few species or to small, relict populations adapted to unique conditions. The idea of emptiness was not absolute but relative, a thinning of life toward the limits of environmental tolerance. Pressure and cold added to the sense of improbability. Although earlier studies had shown that certain deep-sea organisms tolerate high compression, the combined extremes of hadal zones were still poorly understood. It seemed reasonable to predict that only a handful of taxa would possess the biochemical adaptations necessary to maintain cellular function under hundreds of atmospheres. Enzymatic activity, membrane fluidity, and protein structure all change with pressure and temperature. Without abundant energy to offset these constraints, diversity was expected to be low. These assumptions were grounded in logical extrapolation from shallower systems. In many ecological contexts, reduced energy availability correlates with reduced species richness. Deserts on land support fewer organisms than tropical forests. By analogy, the trench was imagined as an oceanic counterpart to arid terrain, vast and quiet, sustaining only scattered inhabitants. Yet, such expectations were formed largely without direct observation. They arose from models, indirect measurements, and limited sampling at depths shallower than the trench itself. The abyss remained mostly unseen, its biological character inferred rather than documented. In the absence of comprehensive data, simplicity seemed plausible. As research vessels prepared to send instruments downward, the prevailing narrative suggested austerity. The deepest Atlantic waters were thought to host a minimal community shaped by constraint, defined more by survival than by abundance. This framework guided early expedition planning and influenced the questions scientists carried with them. Within that calm anticipation lay an image of the trench as a realm where life approached its lower boundary, sustained by faint inputs and adapted to endure rather than to flourish. Long before cameras captured amphipods or sediment cores revealed complex shells, life in the trench was already present at a scale too small for the eye to see. Within the fine grains of mud resting along the deepest Atlantic floor, microbial communities persist in densities that challenge earlier assumptions of scarcity. These organisms do not drift visibly through illuminated water. They inhabit the sediment itself, embedded between mineral particles where pressure and cold remain constant. At depths exceeding 8,000 meters, Compression alters the physical behavior of biological molecules. Proteins can change shape, lipid membranes can stiffen. For surface organisms, such conditions would disrupt cellular function, yet certain microbes, often described as pisophiles, have evolved biochemical systems attuned to high pressure. Their internal chemistry is not merely tolerant of compression, but stabilized by it. One of the most significant adaptations lies within the cell membrane. In typical bacteria, membranes are composed of lipid molecules arranged in a bilayer that must remain fluid enough to allow transport of nutrients and waste. Under extreme pressure, membranes tend to become rigid, impairing movement of molecules across the boundary. Deep sea microbes counter this effect by incorporating higher proportions of unsaturated fatty acids into their membranes. The bends within these molecules prevent tight packing, preserving flexibility even when external force increases. Enzymes, the catalysts that drive metabolism, also require modification. Pressure can distort protein structure, reducing catalytic efficiency. In hadal microbes, subtle variations in amino acid sequences help maintain functional conformations under compression. Some species produce molecules known as piezolites, small organic compounds that stabilize proteins against deformation. Through these adjustments, metabolic reactions proceed at rates sufficient to sustain life in a cold, high-pressure environment. Energy acquisition within the trench sediments reflects the limited but continuous supply of organic matter descending from above. Microbes break down complex carbon compounds that settle into the mud, transforming them through respiration and fermentation pathways. Oxygen penetrates only a short distance into the sediment before being consumed, creating microenvironments where different metabolic strategies dominate. In oxygen-rich upper layers, aerobic bacteria oxidize organic material. Deeper below, anaerobic communities rely on sulfate reduction or methanogenesis, extracting energy from chemical gradients rather than from light. These sediment communities form structured ecosystems on a microscopic scale. Layers only millimeters thick can contain distinct assemblages of organisms adapted to specific chemical conditions. The trench floor is therefore not biologically uniform, but vertically stratified. Each stratum supports microbial populations tuned to pressure, temperature, and available substrates. Genetic analyses of samples retrieved from the trench have revealed a diversity of lineages, some closely related to surface species, others representing branches unique to extreme environments. DNA sequencing indicates that gene clusters associated with membrane fluidity, protein repair, and stress response are particularly abundant. Horizontal gene transfer appears to play a role in spreading advantageous traits among deep-dwelling microbes, accelerating adaptation within this constrained habitat. Despite their microscopic size, these organisms influence broader biogeochemical cycles. By decomposing organic matter and recycling nutrients, they regulate the chemical composition of pore water and overlying seawater. Carbon processed within trench sediments can be stored long-term or released slowly back into the ocean system. In this way, microbial life contributes to processes that extend far beyond the immediate boundary of the trench. The discovery of active microbial communities at such depth reshaped earlier expectations of emptiness. Instead of a sterile basin, the trench contains a dynamic microbial network operating under constant pressure. Invisible to the naked eye, yet measurable through molecular tools, these organisms demonstrate that life's fundamental machinery can adjust to compression that would overwhelm surface-adapted cells. Within grains of sediment, at temperatures near freezing and under immense force, biochemical systems continue their quiet work, sustaining a hidden layer of the Atlantic's deepest ecosystem. They appear first as pale movement against the sediment, small arcs of motion in the beam of artificial light. Amphipods, shrimp-like crustaceans with curved bodies and restless antennae, gather where organic matter settles. In the deep Atlantic Trench, some species grow larger than their shallow water relatives, their forms magnified by an environment defined by pressure and scarcity. These amphipods belong to a group of scavengers adapted to life at extreme depth, recorded thousands of meters below the surface. They occupy a niche shaped by intermittent opportunity. When fragments of organic material descend from above, or when the body of a fish reaches the trench floor, amphipods are often among the first to arrive. Their movements are quick and coordinated, guided by chemical cues dispersed through still water. At hadal depths, their exoskeletons must endure compression far beyond that experienced by coastal species. Rather than relying solely on rigid mineralization, deep-dwelling amphipods incorporate structural features that reduce vulnerability to pressure. The chitinous layers of their exoskeletons are reinforced in patterns that distribute force evenly. Calcium carbonate, common in many shallow crustaceans, is less dominant here, replaced in part by materials that maintain integrity without becoming brittle under compression. Within their tissues, adaptations extend beyond the outer shell. Cellular membranes contain lipid compositions that preserve fluidity despite low temperature and high pressure. Proteins involved in muscle contraction and digestion function efficiently under conditions that would impair related species nearer the surface. These physiological adjustments allow amphipods not merely to survive, but to remain active, swimming short distances and burrowing into sediment. Their feeding behavior reflects the unpredictability of resources. Amphipods are opportunistic scavengers. Sensitive antennae detect dissolved chemicals released by decomposing matter. Once a food source is located, they aggregate rapidly, forming dense clusters around carrion. Observations from remotely operated vehicles have shown dozens of individuals feeding simultaneously, tearing soft tissue with specialized mouthparts. The pace of consumption can be surprisingly swift, reducing larger carcasses to skeletal remains within days. Between such events, amphipods rely on smaller particles embedded in sediment. They sift through fine grains, extracting organic fragments deposited over time. This constant low-level feeding sustains populations during intervals when larger food falls are absent. Energy efficiency is essential. Metabolic rates remain lower than those of comparable species in warmer, shallower waters, conserving resources in a habitat where abundance is episodic. Reproduction in these depths follows a measured rhythm. Females carry eggs within a brood pouch until they hatch, releasing juveniles that resemble miniature adults. Development proceeds slowly in the cold environment, yet survival rates can be sufficient to maintain stable populations. The relative absence of large predators in the trench floor ecosystem may contribute to this persistence, allowing amphipods to occupy a prominent role in benthic communities. Some species display translucent bodies, their internal organs faintly visible under illumination. Others exhibit thicker pigmentation, though color holds little functional meaning in perpetual darkness. Eyes, when present, are often reduced. Sensory reliance shifts toward tactile and chemical detection rather than vision. In the deep Atlantic Trench, amphipods serve as intermediaries between descending organic matter and the broader microbial community. By fragmenting carrion and sedimented material, they increase surface area available for bacterial decomposition. Their activity accelerates nutrient recycling within the hadal environment. Once imagined as nearly empty, the trench floor reveals through these crustaceans a scene of quiet industry. Amphipods move deliberately across the sediment, their curved forms adapted to compression and cold. In their persistent scavenging, they demonstrate how even sporadic resources can sustain visible life at depths once assumed too extreme for anything larger than microbes to endure. In the quiet glow of descending cameras, a small shape drifts just above the sediment, neither hurried nor still. It does not resemble the armored crustaceans that patrol the trench floor. Its body appears soft, almost translucent, moving with a subtle undulation that seems effortless in the heavy water. This is a deep-sea snailfish, recorded at depths approaching the limits of vertebrate life. Snailfish observed near hadal zones inhabit regions once assumed too extreme for bony fishes. Their presence extends into depths beyond 7,000 meters, with some species documented near the deepest sections of the Atlantic Trench. In such places, pressure rises to levels that compress rigid structures and challenge the stability of complex tissues. Yet these fishes persist, their forms shaped by conditions that discourage hardness and favor flexibility. Unlike many shallow water fishes, deep-sea snailfish lack a swim bladder. In surface species, this gas-filled organ regulates buoyancy, allowing precise control over vertical position. At extreme depth, however, gas becomes impractical. Compression would collapse any air-filled cavity, and maintaining internal pressure equal to the surrounding water would require structural reinforcement that is energetically costly. By dispensing with the swim bladder entirely, snailfish avoid this vulnerability. Their bodies achieve near neutral buoyancy through composition rather than internal gas. Much of that composition is gelatinous. The musculature of hadal snailfish contains higher proportions of water and less dense tissue compared to many coastal relatives. This reduces overall body density, allowing the fish to hover just above the seafloor without continuous exertion. The softness that seems fragile in appearance is, in fact, an adaptation. Under immense pressure, flexible tissues distribute force more evenly than rigid ones. The fish's skeletal elements are reduced and lightly ossified, minimizing points of structural stress. At the cellular level, biochemical adjustments parallel those seen in deep-dwelling microbes and invertebrates. Proteins that support muscle contraction and neural signaling must maintain function despite compression. Certain small organic molecules accumulate within snailfish cells, stabilizing protein structure against deformation. These compounds, including trimethylamine N-oxide, increase in concentration with depth, counteracting the effects of pressure on molecular interactions. Vision in these depths offers limited advantage. Snailfish possess eyes, yet they are often reduced compared to shallow water species. The trench environment provides little ambient light, and energy conservation shapes anatomy. Instead, sensory emphasis shifts toward detecting subtle vibrations and chemical cues in the water. The lateral line system, a network of sensory pores along the body, remains sensitive to movement, allowing the fish to perceive approaching prey or the disturbance of sediment. Feeding behavior reflects available resources. Snailfish consume small crustaceans and other invertebrates that inhabit the trench floor. Their mouths are adapted for grasping soft-bodied prey rather than crushing hard shells. Observations from deep-sea missions show them moving deliberately across the sediment, occasionally pausing as amphipods scatter nearby. In this environment, efficiency replaces speed, and energy expenditure remains measured. Reproduction at hadal depths remains less understood, yet egg development and larval dispersal likely occur within similar pressure regimes. Juveniles may occupy slightly shallower zones before descending as they mature, following gradients of pressure and temperature suited to their physiology. The deep-sea snailfish stands as one of the most compelling vertebrate examples of adaptation to crushing pressure. Its gelatinous body and absence of gas-filled organs illustrate a design shaped by constraint. In the dim reach of artificial light, it moves with quiet assurance, demonstrating that even complex skeletal animals can adjust to a world defined by compression, cold, and darkness near the ocean's deepest Atlantic floor. Scattered across the trench floor, partially embedded in fine sediment, lie structures that at first resemble fragments of sponge or abandoned shells. They appear motionless and mineral, irregular in outline, sometimes branching, sometimes flattened against the mud. Yet these forms are alive. They are xenophyophores, among the largest single-celled organisms known, inhabiting some of the deepest regions of the Atlantic. A xenophyophore is not a colony of many cells, but a single cell expanded to remarkable scale. Its body consists of a network of branching strands and chambers enclosed within a delicate organic membrane. Inside, cytoplasm flows slowly around numerous nuclei distributed throughout the structure. This multinucleate arrangement allows the organism to maintain metabolic control across a body that may span several centimeters, an immense size for a unicellular life form. These organisms reside directly within the sediment of the trench floor. They anchor themselves lightly, incorporating surrounding particles into their structure. Fine grains of mineral and fragments of organic matter adhere to the outer surface, giving xenophyophores a mottled appearance that blends with the substrate. The sediment is not merely a resting place, but an integral part of their existence. Through extended pseudopodia, thin projections of cytoplasm, they capture detritus and microscopic particles from the surrounding mud. Growth in this environment proceeds slowly. Energy is limited, and temperature remains near freezing. Xenophyophores expand at a measured pace, adding material incrementally as resources allow. Over time, their tests, the fragile outer frameworks, composed of organic compounds and sediment grains, can reach sizes that dwarf typical protists. Radiocarbon analyses suggest that some individuals may persist for decades, their steady metabolism sustained by a constant but sparse supply of organic debris. Despite their simplicity at the cellular level, xenophyophores influence the structure of the trench ecosystem. Their physical presence alters the texture of the seafloor. By protruding above the sediment surface, they create microhabitats that shelter smaller invertebrates. Amphipods and tiny worms may gather around their contours, using them as refuge or feeding grounds. In this way, a single cell becomes a focal point for localized biodiversity. Internally, xenophyophores accumulate waste products in distinct compartments, isolating potentially harmful byproducts from active cytoplasm. This compartmentalization allows them to maintain function even as sediment particles and absorbed materials build up within their structure. The separation of living cytoplasm from stored residues reflects a form of internal organization that blurs the boundary between unicellular and multicellular complexity. Genetic studies have placed xenophyophores within the foraminifera, a group of protists known for constructing elaborate shells. Yet, unlike many shallow water foraminifera that produce calcareous tests, xenophyophores rely largely on organic frameworks reinforced by sediment grains. This adaptation may reduce vulnerability to dissolution under high pressure and low temperature conditions prevalent in the trench. In the broader context of the deep Atlantic, xenophyophores represent an alternative path to size and influence. Without tissues, organs, or differentiated cell types, they achieve dimensions and ecological roles typically associated with multicellular organisms. Their presence demonstrates that complexity can emerge through expansion of a single cell, given sufficient stability and time. On the quiet floor of the trench, these giant protists rest within the mud, accumulating particles and shaping the immediate landscape. Their slow growth and enduring form contribute to a mosaic of structures across the sediment. In a realm once imagined as biologically sparse, xenophyophores stand as subtle architects, single-celled yet expansive, integrating mineral and living matter into one continuous body, adapted to the deep Atlantic's most secluded depths. Between the sunlit surface and the absolute darkness of the trench floor lies a realm where light is created rather than received. In these mid-water depths, far below the last traces of daylight, living organisms generate their own illumination. Brief flashes shimmer and vanish, cold blue sparks suspended in black water, each one the product of a chemical reaction unfolding within living tissue. Bioluminescence in the Atlantic midwater is widespread. Many gelatinous zooplankton, small crustaceans, and fishes possess specialized organs called photophores. Within these structures, a light-producing molecule known as luciferin reacts with oxygen in the presence of an enzyme called luciferase. The reaction releases energy in the form of visible light, often in wavelengths that travel efficiently through seawater. Blue and blue-green dominate, since these colors propagate farther in the ocean than red or yellow. The distribution of these luminous organisms becomes denser in the twilight and upper midnight zones above the trench. Here, sunlight no longer penetrates, yet life remains abundant enough to sustain complex interactions. In this darkness, light serves as language. A quick pulse may signal distress when a predator approaches. A steady glow along the body of a fish can break up its silhouette, a strategy known as counter-illumination, reducing contrast against faint residual light filtering from above. Predators also exploit bioluminescence. Some species use lures, dangling luminous appendages to attract curious prey. Others detect the flashes emitted by startled organisms and home in on the disturbance. In this environment, light becomes both shield and invitation, tool and vulnerability. Each emission carries information across distances that would otherwise be silent. The chemical basis of this glow is remarkably efficient. Nearly all the energy released in the luciferin reaction converts directly to light rather than heat, producing what is often described as cold light. This efficiency is essential in an environment where energy is limited. Producing illumination without significant thermal waste conserves resources. The molecules involved vary among taxa, yet the underlying principle remains similar: oxidation coupled with enzyme catalysis. Bioluminescent signals also facilitate communication within species. Certain shrimp and squid release luminous clouds into the water, momentarily confusing predators while allowing escape. Some fishes display species-specific patterns of photophores along their bodies, patterns thought to assist in recognition during mating. In a world without ambient light, such displays replace coloration and visual markings common in shallow waters. Above the trench, where descending organic matter continues to drift downward, these luminous organisms occupy a vertical corridor of activity. Their flashes punctuate the darkness intermittently rather than continuously. Long stretches remain unlit, then a brief cascade of sparks appears as one organism disturbs another. The water column becomes a layered system of interactions shaped by chemical light. As depth increases toward the trench floor, the frequency of bioluminescent events declines. Fewer midwater organisms inhabit the extreme hadal zone, and the concentration of life shifts toward the sediment. Yet above, in the vast column between surface and abyss, bioluminescence remains a defining feature of the ecosystem. In perpetual darkness, light acquires heightened significance. It marks presence, conveys intent, and alters behavior. The trench itself rests in shadow, but the waters above it flicker with brief radiance generated by living chemistry. Each flash reflects an adaptation shaped by absence of sunlight, demonstrating that even where the sun cannot reach, illumination can still emerge from within the bodies of those who dwell in the deep Atlantic. Far above the trench, in waters bright with plankton and shifting currents, energy begins a slow descent. It does not travel as beams of light or visible streams, but as fragments. Tiny particles drift downward in a continuous, almost invisible rain. This steady fall is known as marine snow, a mixture of organic debris that links the surface ocean to the abyss far below. Marine snow forms from the remains of microscopic life. When phytoplankton complete their brief cycles near the surface, their cells sink. Zooplankton produce fecal pellets that fall more rapidly, compact and dense. Mucus-like aggregates bind loose particles together, increasing their size and weight. As this material descends, it is consumed repeatedly, broken into smaller fragments, reassembled, and consumed again. By the time it approaches hadal depths, only a fraction of the original organic carbon remains. Yet even that fraction matters. In the deep Atlantic trench, where sunlight never reaches, marine snow provides the primary connection to surface productivity. The rain of particles settles onto sediment, accumulating slowly over time. Microbes process the smallest fragments. Amphipods and other invertebrates sift through the grains, extracting energy from what arrives. This downward flux sustains a baseline of life, modest but persistent. Occasionally, a far larger pulse interrupts the steady fall. When a whale dies and its body sinks through the water column, it delivers a concentrated reservoir of nutrients to the seafloor. These whale falls are rare, yet their impact is profound. The carcass becomes a temporary oasis, supporting dense assemblages of scavengers and decomposers. Amphipods gather quickly, joined by fishes and crustaceans that consume soft tissues. As decomposition progresses, bacteria colonize remaining bones, generating chemical compounds that sustain specialized organisms for years. Such events reveal the layered structure of scavenging networks in the abyss. Initial consumers fragment large material into smaller pieces. Secondary organisms feed on residues and microbial films. Over time, what began as a single large body disperses into sediment, entering the broader detrital cycle. The trench floor, though distant from the surface, is not isolated from its biological rhythms. Energy transfer from surface to depth is neither rapid nor efficient. Most organic production in sunlit waters is recycled within the upper ocean. Only a small percentage escapes this recycling and begins the long descent. Temperature, pressure, and time shape what ultimately arrives. The deeper the basin, the longer the journey, and the greater the losses along the way. Despite this inefficiency, the abyss does not exist outside the planetary system of circulation. Currents transport dissolved organic matter horizontally as well as vertically. Particles carried by storms or seasonal blooms in one region may settle thousands of kilometers away. In this sense, the trench receives contributions from distant ecosystems integrated through ocean-wide movement. Life in the deep Atlantic reflects this reliance on external input. Growth rates remain slow, population densities remain lower than in surface waters. Yet the continuity of marine snow and the occasional arrival of larger falls ensure that energy, however diluted, continues to reach even the most secluded depths. The trench floor therefore represents the terminus of countless journeys. Each fragment that settles has passed through layers of consumption and transformation. What arrives is the final remnant of sunlight-driven productivity, converted into sustenance for organisms adapted to pressure and cold. In this quiet exchange between surface and abyss, the deep Atlantic remains connected to the brighter world above, sustained by a rain of matter that never fully ceases. What returned from the trench was not silence, but complexity written in fragments of tissue and strands of genetic code. The assumption of near emptiness began to soften as sample after sample revealed variation where uniformity had been expected. Beneath layers of cold sediment, diversity persisted in forms both visible and microscopic. Early collections hinted at this richness. Small invertebrates, sorted from mud, displayed subtle anatomical differences that resisted easy classification. Some amphipods carried unfamiliar proportions. Worms exhibited variations in segmentation and bristle arrangement. These distinctions were not dramatic, yet they accumulated, suggesting that the trench floor hosted more than a handful of hardy survivors. The deeper shift in understanding came with genetic analysis. Traditional taxonomy depends on visible traits, yet in extreme environments, morphology can converge, disguising distinct lineages beneath similar shapes. By extracting DNA from collected organisms, scientists began to compare genetic sequences directly. Mitochondrial genes, often used as molecular markers, revealed divergences that physical inspection alone had overlooked. In some cases, two specimens nearly identical in appearance differed significantly at the genetic level, indicating separate species adapted to slightly different ecological niches. Sediment cores yielded environmental DNA, fragments shed by organisms as they moved, fed, or decayed. Even when whole bodies were absent, their genetic signatures remained within the mud. Sequencing this environmental DNA expanded the inventory of life beyond what nets and cameras could capture. Patterns emerged gradually. Species richness did not decline in a simple linear fashion with depth. Instead, certain groups diversified within the trench, possibly isolated over long timescales by topography and pressure gradients. The steep walls and distinct water masses of the hadal zone may act as barriers, limiting gene flow with surrounding abyssal plains. Isolation can foster divergence, allowing new species to arise even under constrained energy conditions. Microbial diversity proved equally surprising. Genetic surveys of sediment communities identified numerous bacterial and archaeal lineages not previously recorded in other marine settings. Some sequences showed only distant similarity to known organisms, hinting at evolutionary branches shaped by prolonged residence under high pressure. The trench, once thought to be biologically sparse, contained a mosaic of microbial specialists adapted to microhabitats within millimeters of sediment. The discovery of unknown species did not occur in dramatic bursts, but through careful accumulation of data. Each expedition added sequences to growing databases. Some organisms could not yet be cultured or fully described, existing only as genetic signatures awaiting further study. Yet their presence was undeniable. Biodiversity in the deep Atlantic trench exceeded early projections, challenging models based solely on energy limitation. This richness does not imply abundance in the way tropical reefs teem with visible life. Population densities remain lower than in sunlit waters, and resources remain scarce. However, diversity refers not only to numbers but to variation. Within the constraints of pressure and cold, evolution has generated multiple solutions, each encoded in DNA and expressed in form and function. The trench floor thus represents a reservoir of distinct lineages, many restricted to extreme depth. Genetic tools continue to refine this picture, revealing relationships and adaptations invisible to the eye. Where once emptiness was anticipated, a layered complexity now appears, subtle yet persistent. In the quiet sediment of the deepest Atlantic, biodiversity unfolds at scales ranging from microscopic genes to delicate invertebrate bodies. Through sequencing and patient observation, scientists have learned that depth does not erase variation. Instead, it reshapes it, producing communities more intricate than early theories allowed, sustained by faint inputs of energy and by evolutionary processes operating in isolation over immense spans of time. In samples lifted carefully from the trench floor, alongside fragments of shell and organic debris, researchers began to notice particles that did not belong to any natural cycle. They were small, often translucent or brightly colored under laboratory light, irregular in shape yet unmistakably manufactured. Even at depths once thought remote from surface influence, traces of plastic had arrived. Microplastics, defined as particles less than 5 millimeters in size, originate from the breakdown of larger plastic items or from microbeads and synthetic fibers released directly into water systems. Rivers carry them to the sea. Ocean currents disperse them across basins. Sunlight and mechanical abrasion fragment larger pieces into smaller ones, which can remain suspended in the water column for extended periods before slowly descending. The journey downward mirrors that of organic matter, yet with a different outcome. Plastic does not decompose in the same way as biological material. Instead, it persists, drifting with marine snow and becoming entangled within sinking aggregates. As these mixed particles descend, some are consumed by midwater organisms. Others settle eventually into the deepest sediment of the Atlantic Trench. Evidence of this descent has appeared within the bodies of amphipods collected from hadal depths. When dissected and analyzed, some individuals contained microplastic fibers and fragments within their digestive tracts. These particles likely entered as amphipods, fed on detritus or scavenged material already contaminated during its passage through upper layers. The ingestion of plastic at such depth illustrates how thoroughly surface activity can permeate ocean systems. Chemical analysis reveals that certain plastics can absorb persistent organic pollutants from seawater, concentrating compounds such as polychlorinated biphenyls along their surfaces. When ingested, these contaminants may transfer to tissues, though the long-term physiological effects in deep-sea organisms remain under study. The trench, once imagined as isolated by distance and pressure, proves connected through circulation and gravity. The presence of plastic in the abyss does not appear as floating debris or visible accumulation. Instead, it is subtle, dispersed among natural grains of sediment, yet its significance lies in what it represents. Material engineered and discarded at the surface has traversed thousands of meters to settle in one of Earth's most extreme habitats. Pressure and cold do not halt this transfer. They simply receive what descends. Sediment cores from hadal zones have revealed microplastic concentrations comparable to or exceeding those in some shallower regions. The trench functions as a terminal sink within the ocean's circulation system. Particles transported across vast distances eventually collect in deep basins where low energy conditions limit resuspension. In this way, the abyss records aspects of human industry within its geological layers. This incorporation of plastic into deep ecosystems complicates earlier narratives of remoteness. While the trench remains physically distant from human presence, it is not insulated from human influence. Through atmospheric deposition, river discharge, and ocean currents, synthetic materials travel far beyond their points of origin. The amphipods that sift through sediment, the microbes that process organic fragments, and the xenophyophores that integrate surrounding particles now interact with substances absent from evolutionary history. The scale of contamination varies, and research continues to assess ecological consequences. Yet the fact of arrival is clear. At the bottom of the Atlantic, beneath immense pressure and near freezing water, fragments of modern life persist alongside ancient sediments. The abyss reflects not only geological processes and biological adaptation, but also the reach of human activity. Even here, in a place defined by depth and darkness, the imprint of the surface world settles quietly into the mud. Far below the stillness of the trench floor, the crust does not rest. Though the sediment above appears undisturbed, the boundary between tectonic plates remains in motion, measured not in sudden leaps of distance, but in accumulated strain. The Atlantic basin is often described as comparatively quiet, yet near the trench, forces converge in subtle and persistent tension. At this boundary, the North American plate interacts with the Caribbean plate in a complex pattern of compression and lateral motion. The movement is neither purely sliding nor directly colliding, but a combination that bends and stresses the descending crust. Over years and decades, stress builds within rock layers that resist deformation. The accumulated energy does not vanish. It remains stored until released as seismic waves that travel outward through the earth. Earthquakes beneath the trench vary in magnitude and depth. Some originate in the upper layers of crust, closer to the seafloor. Others occur deeper, along zones where the descending plate penetrates into the mantle. These deep focus earthquakes can originate hundreds of kilometers below the surface, far beneath the sediment and water above. At such depths, rock behaves differently than at shallow levels. Pressure and temperature alter its physical properties, yet brittle failure still occurs within the subducting slab. Seismic monitoring networks record these events through instruments placed on islands and continental margins. Waves generated beneath the trench travel efficiently through solid earth, detected as minute vibrations, long before any surface expression becomes visible. In most cases, the energy dissipates without noticeable disturbance at sea level. The trench absorbs and reflects this motion, while the overlying water column dampens its immediate effect. The geological instability associated with this boundary reflects a balance between motion and resistance. Plates move only a few centimeters each year, yet over time, this gradual displacement reshapes entire basins. The trench itself owes its depth to prolonged deformation, where one segment of crust has been forced downward relative to another. Each earthquake represents a brief adjustment within that broader tectonic narrative. In certain circumstances, seismic activity can trigger landslides along steep trench walls. Sediment accumulated over millennia may shift suddenly, cascading downward and resettling in deeper sections. Such movements alter local topography, creating new contours on the trench floor. These changes are often subtle when viewed against the vast scale of the basin, yet they reflect ongoing geological dynamism. The deep focus earthquakes beneath the trench offer insight into the internal structure of the descending plate. Their distribution traces the geometry of subduction, revealing how the slab bends and penetrates the mantle. By analyzing seismic wave velocities, geophysicists infer temperature variations and compositional differences within the rock. In this way, earthquakes serve as both expression and probe of geological processes. Despite this activity, the trench remains largely undisturbed at the surface. Ships pass overhead, unaware of the strain released far below. The water above does not ripple with every tremor. Instead, the energy travels invisibly through rock, attenuating as it spreads. Within the quiet darkness of the trench, instability coexists with apparent stillness. Compression, bending, and fracture continue beneath layers of sediment, shaping the basin incrementally. The ocean conceals these movements from immediate view, yet instruments and careful analysis reveal their presence. Beneath the deepest Atlantic waters, the planet's tectonic framework remains active, adjusting slowly, releasing strain and pulses that echo through the solid Earth, while the abyss above remains outwardly calm. Long cylinders of mud, lifted carefully from the trench floor, resemble unremarkable columns of gray and brown. Yet within these cores lies a layered archive, each band of sediment recording conditions that once existed at the ocean surface. Extracted through careful drilling and sealed to preserve structure, these samples offer a vertical timeline extending backward through thousands and sometimes millions of years. As particles descend from above, they settle gradually, forming thin strata. Fine mineral grains mingle with fragments of shells and microscopic skeletons. Over time, additional layers accumulate, compressing those beneath without erasing them. In the trench, where currents are subdued and disturbance is limited, sediment can build in relatively continuous sequences. Each centimeter may represent decades or centuries of deposition. Core sampling allows scientists to read this sequence. By slicing the column into measured intervals, researchers examine variations in composition from top to bottom. Changes in grain size can indicate shifts in current strength or sediment source. Alterations in chemical signatures reflect fluctuations in ocean circulation and productivity. The deeper the layer, the older the record it preserves. Embedded within many layers are fossilized microorganisms, particularly the mineralized remains of foraminifera and other planktonic species. These tiny shells, composed primarily of calcium carbonate, form in surface waters before sinking after death. Their species, composition, and isotopic ratios provide insight into past ocean temperatures and chemistry. Oxygen isotopes within their shells vary according to the temperature and ice volume present at the time of formation, allowing reconstruction of glacial and interglacial cycles. In this way, the trench sediment becomes a climate archive. Variations in microfossil abundance reveal periods of higher productivity linked to nutrient availability. Shifts in isotopic composition mark transitions between colder and warmer global phases. Layers rich in certain minerals may correspond to dust transport from distant continents during arid intervals. Each band contributes to a broader narrative of planetary change. Carbon storage within these sediments also plays a significant role in Earth's long-term balance. When organic matter settles and becomes buried before complete decomposition, its carbon is effectively removed from short-term circulation. Over geological timescales, such burial influences atmospheric carbon dioxide levels. The trench, acting as a terminal basin, accumulates material that may remain sequestered for extended periods. Not all organic carbon survives burial. Microbial communities within sediment continue to process material, releasing some carbon back into dissolved forms. Yet, a portion escapes oxidation, preserved within deeper strata. The balance between decomposition and burial determines how much carbon is locked away. Through core analysis, scientists estimate rates of accumulation and infer the trench's contribution to global carbon cycles. The physical properties of sediment layers can also reveal episodes of disturbance. Turbidites, deposits formed by underwater landslides, appear as distinct layers of coarser material interrupting finer sequences. These events, triggered by seismic activity or slope instability, introduce abrupt changes within the otherwise gradual record. By identifying and dating such layers, researchers link geological processes to sedimentary patterns. In the quiet darkness of the trench, sediment accumulates without awareness of the stories it carries. Each particle that settles adds to a stratified history, extending downward into older epochs. Through careful sampling and analysis, this history becomes legible. Fossilized microorganisms, chemical signatures, and patterns of accumulation together form a chronicle of climate, circulation, and carbon storage. Beneath the deepest Atlantic waters, the seafloor preserves a continuous account of Earth's changing surface, written in layers of mud that record time with patient precision. Far from wind-driven waves and surface tides, a slower movement shapes the deep Atlantic. It is not visible from above, nor easily sensed by instruments that measure only surface drift. This motion unfolds over centuries, guided by differences in temperature and salinity that alter the density of seawater. It is known as thermohaline circulation, a planetary scale exchange linking polar regions to the abyss. In the North Atlantic, winter air cools surface waters until they grow dense enough to sink. As sea ice forms, salt is excluded from the ice crystals and left behind in the surrounding water, increasing salinity and further enhancing density. This heavy water descends in great volumes, forming a mass called North Atlantic deep water. Once submerged, it begins a gradual southward journey along the ocean floor. The descent of this water mass is not a single dramatic plunge, but a spreading flow that follows contours of the basin. It fills deep channels and plains, weaving around ridges and slopes. Because it originates at the surface, North Atlantic deep water carries dissolved oxygen absorbed from the atmosphere. As it moves into greater depth, it transports this oxygen into regions otherwise isolated from direct contact with air. This circulation is part of a broader system, often described as the global conveyor belt. Surface currents, driven by wind, carry warm water northward. There, cooling and increased salinity cause it to sink, completing one segment of a loop that eventually extends into other ocean basins. After traveling through the Atlantic's depths, portions of this water rise elsewhere through slow upwelling, returning eventually to the surface and beginning the cycle anew. Within the trench and surrounding abyss, this circulation influences temperature, stability, and chemical composition. The near-freezing waters that fill the deepest Atlantic are not stagnant. They are renewed, albeit slowly, by the continual formation of dense water at high latitudes. The oxygen delivered by these currents supports aerobic organisms on the trench floor, enabling respiration in environments far removed from sunlight. The movement is subtle. Current speeds in the deep ocean are often measured in centimeters per second. Yet over decades and centuries, even such slow flow reshapes chemical gradients. Nutrients regenerated by microbial decomposition are carried outward. Dissolved carbon dioxide, produced by respiration, disperses through the water mass. The trench participates in this exchange, connected to distant regions through density-driven pathways. Variations in thermohaline circulation can alter conditions within the abyss. During colder climatic periods, increased formation of dense water may strengthen deep flow. During warmer intervals, reduced cooling at high latitudes can diminish sinking rates. Sediment records reflect these shifts through changes in isotopic composition and trace elements, offering indirect evidence of past circulation patterns. North Atlantic deep water is not the only contributor to deep Atlantic currents. Interactions with Antarctic bottom water formed near the southern continent introduce additional complexity. These water masses meet and mix, layering the deep ocean with distinct chemical signatures. The trench, positioned within this layered system, receives a blend shaped by global processes. Although imperceptible to casual observation, the deep Atlantic currents maintain a vital link between surface and abyss. They distribute oxygen, regulate temperature, and transport dissolved substances across vast distances. In the still darkness of the trench, life depends not only on falling organic matter, but also on this steady renewal of water. The currents of the deep Atlantic move with quiet persistence, guided by density rather than wind. Their path encircles the planet, connecting polar chill to equatorial warmth, surface exchange to hidden basins. Within this circulation, the trench is neither isolated nor static. It rests within a slow-moving network that sustains its chemistry and subtly shapes its ecosystem over spans of time measured not in days but in centuries. In a realm where dawn and dusk have no meaning, life reorganizes its priorities. Without sunlight to guide behavior or shape daily cycles, organisms of the trench depend on senses attuned to subtler signals. Darkness here is not an absence to be overcome, but a constant condition around which physiology has evolved. Vision, so dominant near the surface, often recedes in importance. Many species possess reduced eyes or lack them entirely. In their place, other sensory systems expand. Fine hairs and specialized receptors line antennae and skin, capable of detecting minute vibrations traveling through water. A distant movement, the faint displacement caused by another organism, becomes information carried across stillness. Chemical sensing plays an equally central role. Molecules released by decaying material, by living tissue, or by reproductive activity disperse slowly through the water and sediment. Organisms detect these gradients with receptors finely tuned to specific compounds. An amphipod may locate carrion by following a trail of dissolved amino acids. Microbes respond to shifts in oxygen concentration or the presence of sulfate. In this environment, chemistry functions as both map and message. The lateral line system in fishes, a network of sensory pores running along the body, remains sensitive even in complete darkness. Through it, subtle pressure changes are perceived as patterns. Movement is felt rather than seen. For species such as hadal snailfish, these tactile cues provide orientation and awareness within a space where light offers little guidance. Metabolism adapts as well. Cold temperatures and limited food supply favor slower biochemical rates. Enzymes operate efficiently at near freezing conditions, but overall energy expenditure remains restrained. Growth proceeds gradually. Reproductive cycles may extend over longer intervals than in shallow waters. Such pacing aligns with the steady but sparse arrival of organic matter from above. Energy efficiency becomes essential. Organisms allocate resources carefully, investing in structures and processes that yield consistent return. Elaborate pigmentation or complex visual displays are rare where they offer no advantage. Instead, emphasis falls on mechanisms that conserve fuel, from streamlined body forms to reduced skeletal mass. Even movement reflects restraint. Many deep-dwelling animals drift or glide rather than swim continuously, minimizing muscular effort. Cellular adaptations complement behavioral ones. Membrane compositions preserve flexibility under pressure and cold, ensuring transport of nutrients without excessive energy cost. Proteins are stabilized against deformation, reducing the need for repair. Some species accumulate compounds that protect cellular machinery, allowing normal function without constant metabolic correction. Reproduction also reflects energy limitation. Broods may be small but well provisioned, increasing the likelihood of survival in a challenging environment. Development can be slow, yet longevity may extend correspondingly, balancing population dynamics over time. Encounters between individuals may be infrequent, so chemical cues often facilitate recognition and synchronization. In the absence of sunlight, life depends on cues carried through water and sediment, on molecules and motion, rather than color and shadow. The trench ecosystem illustrates how sensory emphasis shifts when light disappears. Organisms become interpreters of vibration and chemistry, responding to gradients that would be imperceptible in brighter realms. This adaptation without sunlight does not signify deprivation but transformation. Evolution has redirected attention from illumination to information embedded in other forms. In the deep Atlantic Trench, energy is scarce, yet not absent. Organisms meet that constraint with efficiency and patience, sustaining communities shaped by darkness, but not diminished by it. Time behaves differently in the deepest Atlantic, not because clocks fail, but because life itself unfolds at a measured rhythm shaped by cold, pressure, and limited nourishment. Movements are deliberate. Growth is gradual. Generations turn over with a patience rarely seen in brighter waters. In the trench, metabolic rates remain low. The near-freezing environment slows biochemical reactions, conserving energy in a place where food arrives irregularly. This reduction in pace influences every aspect of survival. Many organisms grow slowly, adding mass over years rather than months. Shells thicken incrementally. Soft tissues develop with restrained efficiency. Nothing is hurried, because haste would demand energy that the environment cannot reliably provide. Longevity follows from this restraint. Certain deep-dwelling invertebrates exhibit lifespans that extend well beyond their shallow water relatives. Some species of corals and sponges anchored along slopes of the trench may persist for decades or longer, accumulating layers as quiet records of passing time. Even mobile creatures, such as amphipods and fish adapted to hadal conditions, often mature later in life. Delayed development allows them to allocate scarce resources carefully before investing in reproduction. Reproductive strategies mirror this measured existence. Rather than producing vast numbers of offspring with minimal investment, many deep-sea species generate fewer young, each provisioned with sufficient reserves to endure uncertain beginnings. Eggs may be larger relative to body size, containing nutrient stores that support extended development. Brooding behaviors, where present, protect embryos within the parent's body or attached structures, increasing survival in a habitat where encounters are infrequent. Encounters themselves are rare. Population densities in the trench are low compared to coastal ecosystems. Vast stretches of sediment may separate individuals of the same species. Chemical signals disperse slowly, guiding potential mates across distance. In some cases, reproduction depends on synchronized release of gametes, triggered by subtle environmental cues, perhaps linked to seasonal variations in organic matter descending from above. Such coordination ensures that even sparse populations maintain continuity. This slow tempo shapes evolutionary pathways. With fewer generations passing over a given span of years, genetic change accumulates gradually. Adaptations refine existing traits rather than producing rapid diversification. Stability in environmental conditions, particularly temperature and pressure, further reduces selective pressure for dramatic shifts. Instead, lineages persist with incremental adjustments, fine-tuning membranes, enzymes, and sensory systems to meet consistent constraints. Isolation within deep basins can foster divergence over long intervals. When populations become separated by geological features or depth gradients, gene flow diminishes. Over extended timescales, subtle differences may accumulate, leading to distinct species adapted to particular niches within the trench. Such speciation unfolds quietly, without sudden radiations, guided by patience rather than urgency. Predation and scavenging also reflect the slow pace. Movements conserve energy. Ambush and opportunism replace sustained pursuit. Carrion may be consumed gradually by successive visitors, each taking a portion without exhausting effort. Microbial communities decompose organic matter steadily, recycling nutrients at rates aligned with limited supply. The cumulative effect is an ecosystem governed by endurance. Life persists not through abundance or speed, but through careful balance between expenditure and intake. Energy budgets remain tight. Growth and reproduction proceed only when conditions permit. Mortality events may leave gaps that persist for years before recolonization occurs. Within this quiet cadence, the trench maintains continuity across centuries. Organisms live long lives, reproduce sparingly, and adapt slowly, yet effectively. The slow pace does not signify fragility. It reflects harmony with an environment defined by constancy and constraint, where survival favors patience and efficiency above all else. Across the world's oceans, trenches do not share identical histories. Though each marks a boundary where tectonic forces converge, their depths, shapes, and biological communities reflect distinct geological paths. When placed beside the vast Pacific trenches, the Atlantic's deepest depression appears both familiar and uniquely its own. The Pacific Basin contains the most extreme examples. The Mariana Trench descends to nearly 11,000 meters, a depth greater than any point in the Atlantic. Several other Pacific trenches approach or exceed 10,000 meters, formed where dense oceanic plates plunge beneath neighboring plates in long-established subduction zones. By comparison, the Puerto Rico Trench reaches just over 8,000 meters at its deepest point. The difference of several thousand meters reflects variations in plate, age, density, and tectonic configuration. Pacific oceanic crust tends to be older and colder by the time it encounters a subduction zone. Age increases density, and greater density encourages steeper descent into the mantle. This process produces trenches of remarkable depth and pronounced curvature. In the Atlantic, much of the oceanic crust remains comparatively young due to ongoing seafloor spreading along the central ridge. Younger crust is less dense, and large-scale subduction is less continuous. As a result, Atlantic trenches are fewer and generally shallower. Geological structure further distinguishes the two basins. Pacific trenches often align with volcanic arcs, chains of islands or continental margins where rising magma forms volcanoes above subducting plates. These arcs create dramatic landscapes both above and below water. The Atlantic's trench north of the Caribbean lacks an extensive volcanic arc of similar scale. Instead, it reflects a more complex interaction between the North American and Caribbean plates, where compression, lateral motion, and localized subduction combine. The trench's shape is influenced by this hybrid tectonic setting, producing a structure that differs from the sweeping arcs typical of the Pacific. Biodiversity patterns also show contrast. Pacific trenches have been explored more frequently, yielding extensive records of amphipods, snailfish, and microbial communities adapted to hadal depths. Genetic surveys there reveal diverse lineages occupying steep gradients from slope to trench floor. In the Atlantic, fewer expeditions have reached equivalent depths. Early assumptions suggested that shallower maximum depth might correspond to lower diversity. Recent sampling challenges that view, indicating that Atlantic hadal communities possess their own assemblages shaped by isolation and local conditions. Connectivity between trenches may be limited. The vast distances separating Pacific trenches allow regional populations to evolve independently over long periods. In the Atlantic, the singular nature of the Puerto Rico Trench creates a more isolated environment within its basin. Gene flow from other hadal regions is unlikely, encouraging distinct evolutionary trajectories. This isolation can result in species found nowhere else, though comprehensive surveys remain incomplete. Exploration history amplifies perceived differences. Pacific trenches became focal points during early deep-sea research, partly due to their record-setting depths. More submersible dives, lander deployments, and remote vehicle missions have been conducted there. The Atlantic Trench, though closer to major research institutions in some cases, received comparatively less sustained attention. Gaps in sampling leave portions of its slopes and floor insufficiently mapped at biological resolution. Despite these contrasts, both Atlantic and Pacific trenches share fundamental traits. Each hosts life adapted to pressure and darkness. Each records tectonic motion in its structure. Depth alone does not determine ecological richness or geological complexity. Instead, local conditions, circulation patterns, and evolutionary history interact to shape each trench. Placed side by side, the differences illuminate how diverse Earth's deep environments can be. The Atlantic Trench may not rival the Pacific in absolute depth, yet it stands as a distinct chapter in the story of converging plates and resilient life. Exploration does not always follow proximity. The Atlantic borders densely populated continents, major research institutions, and long maritime traditions, yet its deepest trench has received less sustained attention than several Pacific counterparts. The reasons lie not in absence of scientific value, but in patterns of funding, perception, and logistical planning that shape where ships sail and submersibles descend. During the 20th century, many landmark deep-sea expeditions focused on the Pacific. Its trenches, including record-setting depths, drew early interest from oceanographers and navies alike. The dramatic figures associated with nearly 11,000 meters carried symbolic weight, encouraging investment in vehicles capable of reaching such extremes. As technology advanced, established programs continued to return to these well-known sites, building cumulative datasets and long-term research networks. Funding structures often reinforce prior focus. Once a region becomes central to a national or international research agenda, infrastructure grows around it. Specialized equipment, partnerships, and training programs develop familiarity with particular basins. Proposals referencing established study areas may appear less risky, increasing likelihood of support. Over time, this concentration can lead to uneven coverage across ocean basins, even when scientific questions apply broadly. The Atlantic's maximum depth, though substantial, is lower than the deepest Pacific trenches. This numerical difference has sometimes fostered the misconception that the Atlantic is less extreme and therefore less urgent to explore. In reality, depth alone does not determine ecological or geological significance. The Puerto Rico Trench presents complex plate interactions, distinctive circulation patterns, and unique biological communities, yet the absence of record-breaking figures has subtly influenced priorities. Logistical considerations also play a role. Deep-sea expeditions require large vessels, experienced crews, and extended time at sea. Weather conditions in the North Atlantic can be unpredictable, especially during winter months when dense water formation occurs. Storm systems and seasonal variability may compress operational windows, complicating scheduling for research cruises that must coordinate international teams and costly equipment. Additionally, trench exploration demands vehicles rated for extreme pressure. Only a limited number of submersibles and remotely operated vehicles worldwide can safely approach hadal depths. When these assets are allocated to ongoing projects in the Pacific, opportunities for Atlantic missions may be postponed. The expense of mobilizing such systems, including transport, calibration, and support staff, further constrains frequency of dives. Historical narratives have contributed to perception. Early deep-sea milestones, widely publicized, centered on Pacific descents. These stories entered textbooks and public imagination, reinforcing association between extreme depth and the Pacific Basin. The Atlantic, though central to global ocean circulation and climate regulation, remained framed primarily through surface processes and transoceanic travel rather than abyssal exploration. Despite these factors, interest in the Atlantic Trench has grown. Advances in autonomous landers, improved pressure-resistant materials, and international collaboration reduce barriers that once limited access. Genetic sampling and high-resolution mapping reveal that unanswered questions remain abundant. Biodiversity surveys indicate that the trench harbors lineages not yet fully described, challenging earlier assumptions of uniformity. The imbalance in exploration reflects a complex interplay of funding decisions, technological distribution, and cultural focus rather than scientific insignificance. The Atlantic's depth may not rival the most extreme Pacific figures, yet its geological setting and biological communities offer insights unavailable elsewhere. As awareness expands and resources diversify, the trench stands ready for continued study. In the broader pattern of ocean research, emphasis shifts gradually. Regions, once peripheral, become central as new tools and questions emerge. The Atlantic's deepest basin remains part of that unfolding process, shaped less by its inherent mystery than by the pathways through which exploration has historically advanced. The data did not arrive with spectacle. It accumulated quietly, line by line in lab reports, frame by frame, in dim footage retrieved from pressure-resistant cameras. Yet, within those measured observations, patterns emerged that altered earlier expectations. The trench was not defined by absence. It was defined by resilience, expressed in forms more intricate than predicted. One of the earliest surprises came from scavengers. Baited landers lowered into the hadal zone recorded amphipods and other crustaceans arriving in substantial numbers within hours. Their bodies were robust, their movements purposeful. Rather than scattered individuals separated by vast intervals, groups gathered efficiently around organic remains. This suggested not only presence, but active networks of detection and response operating across distance. Chemical cues dispersed through water were interpreted quickly, guiding organisms through darkness toward opportunity. The density of microbial life in sediment cores added another layer of revision. Under magnification and molecular analysis, the mud revealed extensive communities of bacteria and archaea. Cell counts reached levels comparable to or exceeding those of shallower deep-sea environments. These microorganisms were not dormant passengers buried by falling debris. They were metabolically active, cycling nutrients and transforming compounds under high-pressure conditions. Their enzymes functioned within parameters once thought restrictive, indicating biochemical flexibility that expanded known limits of life. Genetic studies deepened this shift in understanding. Environmental DNA extracted from water and sediment samples revealed lineages not easily matched to existing databases. Sequences diverged significantly from known relatives, implying long periods of isolation or adaptation to distinct niches. Even among recognizable groups, such as amphipods and snailfish, genetic variation suggested complex population structures rather than uniform assemblages. Diversity extended beyond visible morphology into the molecular architecture of life. Adaptation complexity emerged as another striking element. Proteomic analyses showed modifications in membrane composition, stabilizing proteins against compression and cold. Osmolytes, accumulated within cells, helped preserve structural integrity under extreme hydrostatic load. Some species exhibited specialized metabolic pathways, allowing efficient utilization of scarce substrates. These traits were not single adjustments, but coordinated suites of changes integrated across physiological systems. The presence of thriving scavengers also indicated effective energy transfer from surface to depth. Carrion events, whether from sinking fish or larger vertebrates, supported bursts of activity. Microbes initiated decomposition, releasing compounds that attracted invertebrates. Larger organisms fed, then departed, leaving behind remnants for successive consumers. This cascading use of resources reflected an organized community rather than isolated individuals surviving by chance. Scientists had anticipated survival at the margins. What they encountered instead was structured persistence. The trench floor supported organisms interacting through chemical signals, reproductive strategies, and ecological roles. Microbial mats influenced sediment chemistry. Invertebrates shaped the distribution of organic matter. Even rare fish contributed to trophic connections, extending upward and outward. Perhaps most unexpected was the realization that depth alone did not simplify ecosystems. Complexity persisted despite pressure and darkness. Life did not reduce to a few generalized forms. It diversified within constraint. The hadal environment imposed limits, yet within those boundaries, evolution crafted specialized solutions. Each new dataset reinforced this impression. Imaging, sequencing, and biochemical assays converged on a consistent theme of adaptation interwoven across scales. From microbial cells to mobile scavengers, organisms exhibited features tuned precisely to their surroundings. The trench was not an outlier of biological minimalism. It was a laboratory of endurance, where life expressed ingenuity under compression. The surprises were not dramatic revelations, but accumulations of evidence reshaping understanding. The deepest Atlantic proved neither barren nor simplistic. It revealed a community, sustained by subtle interactions, intricate physiology, and genetic variation that spoke of long residence in the dark. Habitability was once imagined as a narrow band, a delicate balance of warmth, light, and moderate pressure. Early definitions centered on surface conditions, on environments where liquid water, sunlight, and familiar atmospheric chemistry overlapped. The trench complicates that picture. It reveals that life persists far beyond those earlier assumptions, reshaping the boundaries of what is considered possible. At depths exceeding 8,000 meters, pressure surpasses 800 atmospheres. For many decades, laboratory experiments suggested that complex cellular processes would fail under such compression. Proteins were expected to lose shape, membranes to stiffen beyond function. Yet organisms dwelling in the habitable zone demonstrate otherwise. Their molecular structures remain stable, not through resistance alone, but through adaptation. Lipid membranes incorporate specific fatty acids that preserve fluidity. Enzymes exhibit conformations resistant to deformation, maintaining catalytic efficiency under force that would disrupt surface species. These observations expand pressure tolerance limits beyond prior models. Instead of a fixed upper boundary, the threshold becomes dependent on evolutionary history and biochemical adjustment. What appears extreme from one perspective becomes routine for organisms shaped by that environment. The trench thus serves as evidence that physical constraints are not absolute barriers, but variables to which life can respond. Biochemistry within the abyss reveals flexibility in energy acquisition and metabolic pathways. Microorganisms utilize substrates derived from sinking organic matter, from chemical reactions within sediment, and from dissolved compounds transported by deep currents. Some rely on anaerobic processes, using alternative electron acceptors in the absence of abundant oxygen. Others couple reactions that release modest amounts of energy, sufficient when consumption is minimized by slow metabolism. Such versatility informs broader discussions of extreme life. If cellular machinery can operate under immense pressure and persistent cold, it may also function in subsurface oceans or beneath icy crusts elsewhere. The trench provides a terrestrial analog, a natural laboratory demonstrating that life does not require sunlight or mild conditions to endure. Instead, it requires stable liquid water, energy gradients, and chemical building blocks, even when those gradients are subtle. Redefining habitability involves recognizing that environmental constancy can substitute for abundance. The habitable zone offers little variation in temperature, little disturbance from seasonal change. This stability allows organisms to fine-tune physiology to a narrow range, optimizing survival within constraints. Rather than thriving, despite harshness, life persists because it aligns precisely with those conditions. The implications extend to planetary science and astrobiology. Worlds once dismissed due to high pressure or absence of surface light may warrant reconsideration. If Earth's deepest oceans support complex ecosystems, then similar settings elsewhere could be candidates for life, provided chemical energy is available. The trench encourages humility in defining limits. Pressure tolerance, biochemical resilience, and metabolic adaptability converge to challenge earlier frameworks. Habitability becomes less about comfort and more about compatibility between environment and organism. The essential factors shift from surface warmth to internal equilibrium, from sunlight to sustained energy flow. In the deep Atlantic, life does not stretch beyond its means. It inhabits a niche carved by physics and chemistry, demonstrating that boundaries expand when examined closely. The trench stands as a reminder that definitions grounded solely in surface experience capture only a fraction of Earth's possibilities. By existing where pressure is immense and light absent, deep organisms broaden the map of where life can be found and where it might yet emerge under distant seas. Beneath the shifting weather of the surface world, The trench participates in processes measured not in seasons, but in centuries. It does not generate storms or shape daily forecasts. Instead, it functions quietly within Earth's broader climate system, contributing to cycles that unfold far beyond human timescales. One of its most significant roles lies in carbon burial. Organic material produced near the surface sinks gradually through the water column. Some of it is consumed before reaching the seafloor, yet a fraction settles into the trench and surrounding abyssal plains. Within these sediments, carbon becomes incorporated into layers that may remain undisturbed for thousands to millions of years. Microbial communities transform portions of this material, but not all is returned to circulation. Burial effectively removes carbon from short-term exchange with the atmosphere, influencing long-term concentrations of greenhouse gases. The trench's great depth also contributes to deep ocean stability. Water masses filling the abyss are cold and dense, formed in polar regions before descending. Once in place, they change slowly. Temperature fluctuations at the surface rarely penetrate to a dull depths. This stability creates a reservoir insulated from rapid atmospheric variation. In climate terms, such insulation moderates extremes by storing properties that would otherwise shift more abruptly. Heat storage represents another dimension of this system. The ocean as a whole absorbs a substantial portion of excess heat from solar radiation and greenhouse forcing. While most warming is detected in upper layers, a portion is transported downward through circulation. Deep waters accumulate heat gradually, buffering surface changes. The trench, as part of the deepest oceanic domain, participates in this redistribution. Even slight increases in deep water temperature reflect vast quantities of absorbed energy due to the enormous volume involved. Over geological time, interactions between tectonics and climate further connect the trench to planetary regulation. Subduction processes recycle oceanic crust, rich in carbonates and organic matter, back into the mantle. Volcanic outgassing elsewhere eventually returns some of this carbon to the atmosphere, completing a cycle that spans millions of years. The trench marks a boundary where surface-derived materials begin that descent. Although this process is slow, its cumulative effect influences atmospheric composition across epochs. Sediment records within the trench preserve evidence of past climate states. Variations in isotopic ratios, microfossil assemblages, and mineral composition reveal shifts in ocean temperature and productivity. These archives allow reconstruction of glacial and interglacial cycles, linking deep ocean conditions to global climate patterns. The trench thus serves not only as participant, but also as recorder. Deep ocean stability contributes to predictability in long-term climate regulation. Because abyssal waters mix slowly, they dampen rapid changes. This inertia can delay full expression of surface warming or cooling, extending adjustment periods over decades to centuries. While such buffering does not prevent change, it moderates its pace, distributing effects across time. Carbon burial within hadal sediments also interacts with oxygen availability and microbial metabolism. When organic matter is stored rather than fully oxidized, atmospheric oxygen and carbon dioxide levels adjust accordingly. The balance between burial and remineralization influences global biogeochemical cycles. Though individual trenches represent small fractions of the ocean floor, collectively they form part of a network regulating these exchanges. In this way, the trench operates as a quiet stabilizer. It stores carbon, retains heat, archives climate history, and participates in tectonic recycling. Its influence is diffuse rather than dramatic, integrated into systems that govern Earth's long-term equilibrium. Far below familiar horizons, these processes continue steadily, shaping climate not through sudden events, but through persistence measured across deep time. Maps suggest completion. Lines trace contours across the seafloor, color gradients imply precision, and depth values appear settled. Yet beneath those representations lie vast stretches only broadly outlined. Even within the trench itself, fine-scale topography remains incompletely resolved. Multi-beam surveys provide general structure, but steep walls and narrow depressions conceal details that await closer passes. Shadows in acoustic data mark places where resolution fades, reminding researchers that mapping is an ongoing process rather than a finished task. Unmapped areas extend beyond shape into composition, sediment thickness, mineral distribution, and small-scale faulting patterns are known only from scattered cores and seismic profiles. Each sample offers insight into a single point, while adjacent areas may differ in subtle ways. Without dense coverage, continuity between observations must be inferred. The trench floor, though charted in outline, still holds microhabitats whose boundaries remain undefined. Biological knowledge shares similar gaps. Genetic surveys reveal sequences unmatched in reference databases, implying species not yet described. Many organisms captured in imagery or sediment samples are represented by fragments, larvae, or environmental DNA rather than complete specimens. Formal identification requires careful morphological and molecular comparison, a process that unfolds gradually. For every recognized species, others likely persist beyond current sampling effort. The distribution of life within the trench is also incompletely understood. Baited landers attract mobile scavengers, but less mobile organisms may remain underrepresented. Sampling bias shapes perception. Regions visited repeatedly accumulate records, while unvisited slopes and deeper pockets remain quiet in databases. Absence of evidence does not equal evidence of absence, particularly in environments where access is limited. Technological constraints define much of this uncertainty. Vehicles capable of reaching hadal depths are few. Each deployment demands careful engineering, pressure testing, and logistical coordination. Battery capacity, tether length, and communication bandwidth restrict duration and range of observation. Even the strongest materials face limits under prolonged exposure to extreme hydrostatic pressure. Maintenance cycles and cost considerations further constrain frequency of missions. Imaging in total darkness depends on artificial light, which can influence behavior. Some species may retreat from illumination, leaving their presence undetected. Acoustic sensors offer broader coverage but lack fine biological detail. Sediment cores retrieve discrete columns, yet disturb surrounding structure. Every tool reveals certain aspects while obscuring others. Knowledge advances incrementally as methods improve. Environmental variability over time introduces another layer of unknown. Seasonal patterns in organic matter deposition, episodic events such as large carcass falls, and shifts in deep circulation may alter community composition. Long-term monitoring at hadal depths remains rare, limiting understanding of temporal dynamics. Without continuous records, trends must be reconstructed from intermittent snapshots. Future expeditions aim to address these gaps. Advances in autonomous vehicles promise longer endurance and broader mapping coverage. Improved pressure-resistant electronics may allow extended operation near the seafloor. Expanded genetic libraries will help match environmental sequences to known lineages. International collaboration can distribute costs and share expertise, increasing opportunities for sustained exploration. Yet, even with enhanced technology, complete knowledge may remain elusive. The trench is not static. Geological processes continue, sediments accumulate, and biological communities evolve. Each answer reveals further questions embedded within complexity. Scientific inquiry proceeds not toward finality, but toward deeper resolution. What remains unknown does not diminish what has been learned. Instead, it frames the trench as an active field of study, where discovery continues in measured steps. Beneath the mapped contours and recorded depths lies a landscape still unfolding in detail, inviting patient observation and steady return. The surface of the Atlantic rarely hints at what lies beneath. From a distance, it appears continuous and familiar, its tides rising and falling against shores that have shaped human history for centuries. Ships trace steady routes across it. Satellites measure its height to the centimeter. Weather systems sweep across its surface in patterns that feel known. Yet far below that restless skin, the ocean opens into a domain that remains largely unseen. The scale is difficult to hold in mind. Nearly 5 miles of water separate sunlight from the trench floor. Above, storms form and dissipate within days. Below, sediments settle grain by grain across spans that outlast civilizations. The vertical distance alone contains gradients of pressure, temperature, and chemistry that define entirely different ways of existing. What appears unified from above becomes layered and varied with depth. Within this depth, Earth's hidden systems continue their quiet work. Dense waters formed in polar cold slide slowly through the abyss, carrying oxygen and dissolved carbon across basins. Microbial communities transform organic remnants into chemical forms that circulate through sediments and currents. Tectonic plates converge and shift, shaping the trench over geological time. None of these processes announce themselves at the surface, yet each participates in maintaining planetary balance. Exploration has revealed fragments of this concealed world, yet those fragments remain partial. Instruments descend, collect data, and return. Cameras illuminate brief circles of seafloor before ascending again. Samples arrive in laboratories, offering glimpses into adaptations refined under pressure and darkness. Each expedition expands understanding, but also underscores how much remains beyond direct observation. There is a certain humility in this recognition. Despite centuries of navigation and decades of technological advancement, vast portions of Earth's ocean persist beyond detailed knowledge. The familiar horizon hides an environment shaped by forces rarely experienced directly. What seems ordinary from shore conceals extremes of compression, cold, and biological ingenuity. This contrast between surface familiarity and abyssal remoteness reframes perception. The ocean is not merely a backdrop for human activity or a route between continents. It is a layered system extending far below visible waves, containing ecosystems that operate according to principles distinct from coastal waters. Life thrives there not through abundance of light, but through efficiency, resilience, and intricate chemical exchange. The trench, in particular, represents one of the least seen ecosystems on the planet. Its inhabitants live beyond routine reach, shaped by conditions that challenge conventional expectations. Yet they persist, interacting through subtle cues and steady cycles. Their existence widens the sense of what Earth contains, reminding observers that complexity extends into places once assumed barren. Scale, in this context, is both spatial and temporal. Depth measures distance, while sediment layers record epochs. Processes unfold slowly, often imperceptibly, yet accumulate into patterns that influence climate, circulation, and evolution. The trench participates in these broader systems without spectacle. Its influence is distributed, integrated into the planet's ongoing equilibrium. Standing at the edge of the Atlantic, one sees only the surface expression of a far deeper story. Beneath the shifting light and familiar tides lies a frontier defined not by emptiness, but by quiet continuity. Exploration has illuminated portions of that realm, revealing adaptation and persistence under conditions once thought prohibitive. There is no sudden revelation in acknowledging this depth, only an expanded awareness. The ocean remains what it has always been, vast and layered. Within it, the trench rests as a reminder that even well-known places can conceal extraordinary worlds, sustained in silence beneath everyday waves.